Hello and welcome to the September The Nutritionist 2019. I'm Marianne Fessenden from AMTS. We're glad to be back with you for a new webinar this month. For those of you who are joining us for the first time, our format is to deliver a recorded presentation at 2 times on the webinar day, with the presenter joining at the end of each session to answer questions from the listening audience. I am also pre-recording the introduction and ending credits to minimize risk of technical difficulties. These educational webinars are multinational. We have several co-hosts who join to deliver the presentations translated into their country's language and to field questions from their countrymen. Paula Turillo from Argentina has been a constant contributor since the beginning of the series in 2015 and assists in planning the webinars. Elena Bonfante of Dairy Innovations Italia in Italy is a regular co-host, as is Sean Lee of AnsciTech in China. Our potential panelists include Vadim Bekchebnikov of Novo Lab, Russia, and Marcelo Ramos of 3R Labs, or Marcos Neves Pierre of Universidade Federal de Lavras in Brazil. Depending on how you are listening, you can submit queries through me or one of my attending co-hosts. Later, a complete recording of archived webinars as well as a question and answer session for each will be available on the AMTS website. For those of you who would listen to the presentation whilst driving, we have converted the videos into MP3 files that can be downloaded to your device for offline listening. Those podcasts can be found at the Ag Model Systems website under the Webinar tab or Resources tab. We are very pleased and honored to have Dr. Tom Jenkins here today. This is his second presentation of The Nutritionist. Dr. Jenkins attended Penn State University for his BS and MS degrees and received a PhD at Cornell University. After a postdoc at The Ohio State University, he then moved to Clemson University, where he continued to work on dairy cattle nutrition for over 30 years. Dr. Jenkins taught undergraduate and graduate courses in nutrition and dairy cattle, including basic work on rumen lipid metabolism. He has published extensively in scientific journals and conference proceedings and has given numerous invited presentations across more than a dozen countries on lipid metabolism in dairy cattle and the practical aspects of fat feeding. Dr. Jenkins has received numerous awards from Clemson University and the American Dairy Association for his research accomplishments in rumen lipid metabolism. As he will tell you, he has retired, but I'm sure that does not mean that he has more time. Today, Dr. Jenkins will talk about nutritional decisions that cause microorganisms in the rumen to drive down milk fat. Tom is joining us remotely from South Carolina. Remember to jot down any questions you have during the presentation and type them in the Q&A window. I will moderate your questions during the question and answer period.
My name is Tom Jenkins. I'm retired currently from the Department of Animal and Veterinary Sciences at Clemson University in Clemson, South Carolina. And it is indeed my pleasure to be a part of this very successful and important webinar series. So I want to give very much thanks to Dr. Tom Tlucki and for his colleagues, especially Marianne Fessenden, for all the great work that they have done in bringing an educational component to dairy nutrition. So I'm thankful to have the opportunity today to share my thoughts on this subject shown here and talk about the connection between microorganisms in the rumen and how they can influence milk fat and the nutritional decisions you make that impact that connection. Now, there are a multitude of factors that affect milk fat content. We can group them into two general categories of nutritional factors as well as non-nutritional factors. They're both very important. Non-nutritional factors include genetics, stage of lactation. In the first 90 days or so, milk fat will be considerably higher because of mobilization of body fat stores. Season, all areas of the country see cows go through summer slump where milk fat and protein go down. parity, breed, and disease. Now, while these are all very important influences on milk fat content, it's not our focus today. We want to focus on nutritional factors and fill in these question marks. What are the nutritional factors that influence milk fat? We are aware of multiple nutritional risk factors that affect milk fat. These include feeding too much fat, high starch, low rumen pH, wild yeast in fermented feeds, corn silage, high moisture corn, feed delivery, how we mix TMR, how many times we feed a day, how often we push up, feed facilities, do we have adequate bunk space, are we overcrowding, and the presence or absence of ionophores. And when we have low milk fat or it drops down suddenly, we work our way through these list of risk factors, trying to take care of them and get it back to where we think it should be. Oftentimes that's a frustrating process that occurs very slowly, or there's very little movement at all. Now there's surely multiple reasons why that occurs. One of them is oftentimes our impatience, not giving enough time for our changes to take effect and express themselves. But another reason is the fact that these are not independent events, that they all interact with each other, so that a small change in one of these risk factors can impact one or more of the other ones. And that can make it very, very difficult at times to move milk fat in the right direction. I've been searching for an explanation of what explains the interaction that occurs. And again, I'm sure there are a number of explanations, but I think recent results from the Rumen microbiology literature has offered an explanation of what the microorganisms see in the rumen when all these complexity of nutritional risk factors all come together and interact with each other. And that's what I would like to focus on today, to build a case for the explanation for these risk factors from the standpoint of the rumen microbial population. And before we get into that, I want to briefly review the process of mammary synthesis of milk fat. It'll help to see that connection with the rumen. Now, milk fat is nothing more than a collection of individual fatty acids. Fatty acids are long chains of carbons connected by bonds. And it can be as few as 4 carbons in the chain, the way I have drawn it here, all the way up to 18 carbons in the chain. And everything in between, 4, 6, 8, 10, so forth, all the way up to 18. Milk fat is a collection of those fatty acids. Now, where does the mammary gland get these? All these individual fatty acids. One portion of them, it synthesizes on its own in a process called de novo fatty acid synthesis. And in de novo synthesis, it uses acetate from carbohydrate fermentation in the rumen. The acetate gets into the blood, makes its way to the mammary gland, and the acetate along with some butyrate are put together, 2 carbons at a time, to synthesize fatty acids going from 4 carbons all the way up to 16. It stops at 16, can't go any higher. So already you can see the connection between the rumen and the mammary gland. The rumen supplying VFAs or acetate in order to fuel this process of de novo fatty acid synthesis. Now, we said the milk fat has 18 carbons. Where do they come from? Because de novo is not supplying them. The alternative route is for them to come from the blood. The 18 and part of the 16 carbon fatty acids that come from the diet or mobilized from body fat are in the bloodstream, and the mammary gland extracts them out of the blood and incorporates them into these fatty acids. So that gives then 3 fatty acid fractions that we find in milk fat. One group we call de novo, which have less 16 carbons in the chain. The other group is preformed, which are 18 carbons in the chain. And then we have the other group called 16, which are mixed. We're calling mixed because part of the 16 comes from preform extracted from the blood, and part of them is de novo synthesized from acetate in the rumen. That completes all 3 fatty acid fractions that are used to build up milk fat and get us where we want to be. So it takes all 3 of these fatty acid fractions to build up milk fat. Let me give you one example here. This was taken from a paper published in 2017 for cows that had about 4% milk fat content. Now, 0.97 or about 1% of that 4 was de novo, up to 16. 1.4% of it was preformed, that was extracted directly from the blood. That left 1.63 that was mixed. The mixed is just C16. Preformed are 18, and the de novo are less than 16. So remember now that about half of the mixed come from de novo and half come from preformed. So if we split those out and move half of the C16 in de novo, move half into the preformed, that would mean that in this example, where we have about 4% milk fat, roughly 1.8 out of the 4 came from de novo and 2.2 came from preformed. So about half, roughly about half of the milk fat is de novo and half is preformed. Both are substantial contributions to the synthesis of milk fat, which a mammary gland could not do without, either one of them, for us to even come close in our expectations for what our milk fat should be. So the rumen plays a central role in the synthesis of milk fat. We've already talked about how the rumen supplies VFAs in the form of acetate and butyrate to the mammary gland for de novo milk fat synthesis. But the rumen can also provide inhibitors, synthesized inhibitors that get into the blood, go to the mammary gland, and inhibit de novo. And if it's inhibiting de novo, will negatively impact half of the milk fat that's synthesized, and that can have a dramatic impact on the amount of milk fat produced per day and on milk income checks. So if the mammary gland is the engine that is driving the synthesis of milk fat, then I see the rumen as the gas pedal and the brake. Step on the gas and you're supplying more acetate supplied to the mammary gland to make de novo. Step on the brake and you're supplying more inhibitors to shut down the machine, slow it down so it's not carrying on the process of de novo as completely or as thoroughly as you hope it would. Here's an example of stepping on the gas. This is data taken from a Journal of Nutrition article in 2017, where increasing grams of acetate were infused directly into lactating dairy cows and milk fat monitored. You can see with more and more acetate, there's a steady rise in the concentration of milk fat. So supplying more acetate, stepped on the gas, applying more fuel to the mammary gland machine to synthesize milk fat. Now, you don't have to infuse acetate for this to happen. You simply need to have enough fermentable carbohydrate for the cows in order to supply this acetate. Here's an example of stepping on the brakes. In this case, cows received diets that had no added fat or 5% added soybean oil. You can see the fat percent on the control with no added fat was 3.53%. That gave a milk fat yield of 1.12 kilograms per day. Now, when 5% soybean oil was present in the diet, milk fat yield dropped from 3.53 to 2.73, dramatic downturn in milk fat. And likewise, milk fat yield went down significantly from 1.12 to 0.85. So when we looked at the milk fatty acid fractions, okay, this is the changes in the fractions relative to the control. The percent change from the control. De Novo went down 55% when soybean oil was added. Mixed went down 61%. There was a 17% increase in preformed, but it wasn't near enough of an increase to counter the drops in mixed and de novo. Consequently, milk fat went down. This is all the result of the soybean oil causing the synthesis of inhibitors in the rumen. So the question is, where do these inhibitors come from? We certainly want to prevent these from accumulating in the rumen and making it to the mammary gland. They're the primary reasons why we're not achieving the level of milk fat that we want in most cases. So we want to talk about where they come from and what we can do to prevent their synthesis. The source of the inhibitors can be described in a single word: biohydrogenation. In the simplest terms, this is an enzymatic pathway that's carried on by a select group of microorganisms that inhabit the rumen contents. And like all enzymes pathways, its function is to take a substrate and convert it to end product. So the substrate for biohydrogenation are unsaturated fatty acids that are found in the feed of the animal. And these are converted to the end products, which are saturated fatty acids. So the feed mostly has unsaturated fatty acids, but what comes out of the rumen is mostly saturated fatty acids. Let me give a specific example. One of the primary unsaturated fatty acids consumed in typical dairy cow rations is linoleic acid. Linoleic acid has 18 carbons and 2 double bonds in its chain. The 2 double bonds is what makes it an unsaturated fatty acid. The enzymes from biohydrogenation get rid of these 2 double bonds right here and convert it to stearic acid with 18 carbons in the chain, but no double bonds. So it goes from an 18,2 to an 18,0, which converts it from unsaturated to saturated. Now, it's believed that this process is very important to the rumen because linoleic acid, like other unsaturated fatty acids, can have antimicrobial effects that interfere with the growth and function of the microorganisms. So this is a protective mechanism to try to allow for better metabolism and better health of all the microorganisms in the rumen. So biohydrogenation has intermediates. It doesn't go directly from what's in the feed, like linoleic acid, directly to stearic acid that comes out of the rumen. There are intermediates. In fact, there are 2 classes of intermediates. Linoleic acid is first converted to what we call CLA. Then the CLA is converted to trans fatty acids, and then they go to stearic acid. The CLA stand for conjugated linoleic acid, And this is the group of intermediates in biohydrogenation that inhibit de novo milk fat synthesis, these CLAs. And to make a CLA, the microorganisms are taking linoleic acid, just moving the double bonds around to different positions, creating new compounds that were present in the feed, but now some of these are able to inhibit milk fat synthesis and specifically the de novo fraction. So the CLAs in biohydrogenation is a source of inhibitors that put on the brakes that cause de novo to go down and keep our milk fat down. Now, biohydrogenation is very complex. There are 5 major fatty acids coming in the feed. 16O or palmitic acid, 18O is stearic, 18.1 is oleic, 18.2 linoleic, 18.3 linoleic. 2 of them are saturated, palmitic and stearic. I X'd them out because the intermediates, the CLAs, are not made because of the saturated fatty acids. Only the unsaturated fatty acids will contribute to the source of the intermediates in biohydrogenation. Now, there's not one intermediate, there are dozens of them. You can find a multitude of CLAs and a multitude of trans fatty acids being produced as a result of biohydrogenation. But we concern ourselves with only a few that we have identified in past years as being inhibitors of de novo synthesis in the mammary gland. Keep in mind, the biohydrogenation is very complex, yielding a wide variety of intermediates through a complex array of pathways. But we focus on the pathways that generate just the inhibitors that lower milk fat. So we focus on 2 major pathways. Linoleic acid, normally in the predominant pathway, has the double bonds move from the 9 and 12 positions, as shown here, to the 9-11 position. It moves the double bonds closer together, and when it does that, it converts a double bond from cis to trans. This is now a CLA called rumenic acid, and actually it has been shown to have a number of positive health benefits to humans, including anti-atherogenic and anti-carcinogenic properties that are still being looked at and explored. And then the microorganism in the enzymes of biohydrogenation get rid of the cis-9 double bond, so all you have is a trans-11 remaining. That's called vaccinic acid. They do that by adding hydrogen to the double bond to eliminate it. Then in the final step, add more hydrogen, and you have stearic acid as the end product. A minor pathway is, instead of moving the double bond at the 12 position to 11, other enzymes produced by the microorganisms can take the 9 double bond and move it to 10. They move it closer together. When they move it, it converts it from cis-9 to trans-10, and we've generated a new CLA, trans-10 cis-12 CLA. And this is the specific one, this is the inhibitor that we're talking about that can lower milk fat. This is an inhibitor that we don't want our nutritional decisions to cause it to go up in the rumen. And then this CLA, the enzymes of biohydrogenation add hydrogen, converts it to trans-10, gets rid of the cis-12, add more hydrogen, and it goes to stearic acid. This is the results from a study that was done at Cornell University and published in 2004, showing the potency of the trans-10 cis-12 CLA. Just a few grams of it were infused in dairy cow each day. And there were dramatic decreases in milk fat yield. Here. You can see as the trans 10 cis 12 infusion goes up, there's a drop in the decrease in milk fat yield. And it only took a few grams of this COA to give a 20 or 25% drop in milk fat yield, which would cause severe economic problems on a dairy. So, where did the inhibitors come from that we're trying to avoid? They come from biohydrogenation. What part of biohydrogenation? The intermediates. What intermediates? The COAs. Are all COAs the same? No. Some are good, like rumenic acid. There are others that inhibit de novo synthesis in the mammary gland. So we're concerned now in what nutritional signals that we're responsible for as nutritionists that cause things to shift in the wrong direction. Now, I'm not going to refer to these pathways so much by their COA, but by their trans identity. So the trans 10 pathway gives the COA trans 10 cis 12 that causes milk fat to go down. The trans 11 pathway is the one that makes rumenic acid that does not cause problems with milk fat. It causes positive health benefits. So the trans 10 pathway is what we're trying to avoid because it yields the COA that causes milk fat to go down. So your feeding decisions can get you to a crossroads. A crossroads that can determine which way is your linoleic acid going to go. Is it going to go down the trans 11 pathway and produce the COA rumenic acid that does not cause problems with milk fat? Or is it going to go down the trans 10 pathway that produces the T10 C12 trans 10 cis 12 COA that is the known inhibitor of milk fat? That's what we want to focus on. What causes linoleic acid to go down the wrong road in this picture? One of the situations that we know causes linoleic acid to go down the trans 10 pathway or the wrong direction is high unsaturated fatty acids. I'm going to refer to these as RUFL. In fact, scientific investigations that want to intentionally cause milk fat to go down will do it by adding high amounts of unsaturated fat sources like soybean oil, for instance, as a means to reliably cause milk fat to go down. Now, RUFL stands for rumen unsaturated fatty acid load. It's simply the sum of the three major unsaturated fatty acids that a cow consumes every day: oleic acid, linoleic acid, and linolenic acids. If you're feeding mostly corn silage-based TMR, corn and soybean meal, linoleic acid, or 18-2, will by far be the biggest contributor to the RUFL each day. If you're feeding significant quantities of hay or haylage, that will increase the linolenic acid contribution at the expense of 18-2. And oleic acid will go up if you're feeding significant quantities of canola products or tallow, or even some bypass fats now are bringing in oleic acid. I could refer to these simply as unsaturated fatty acids, but I use the term RUFL to refer mostly to load, because the microorganisms don't care whether the unsaturated fatty acids are coming from added fat or from the basal diet. They're having the same effect. So the RUFL, or the load, refers to the quantity of these unsaturated fatty acids that are coming from all sources. And I think the root cause of what causes linoleic acid to go down the wrong pathway in the rumen has to do with too much RUFL interfering with the microbial cell. And remember, and these are results that I did from some analysis that I did in my lab over the years. There is RUFL in everything we put in front of the cow. The only exceptions would be water and minerals. Okay. So everything from corn silage to hay to oil seeds and everything in between contribute RUFL to the rumen environment. I'm going to use distiller's grain, shown here in yellow, as a reference point. If the distiller's grain had 8.6% total fatty acid, the RUFL in this example was 6.8%. So it took 3.2 pounds of distiller's grain dry matter in order to supply 100 grams of RUFL. The pounds of dry matter to supply 100 grams of RUFL is shown in this column. 3.2 pounds of distiller's dry matter to give 100 grams of RUFL. Now, corn silage supplies RUFL. It took 12 pounds of corn silage dry matter to get the same amount of RUFL as 3 pounds of distiller's dry matter. And we feed a lot of corn silage, so it's not difficult to bring in significant quantities of RUFL with corn silage. Alfalfa hay was 2.4% RUFL in my analysis. So it takes 9 pounds of alfalfa hay dry matter to give 100 grams, or the same amount of RUFL as 3 pounds of distiller's grain. Corn grain, 3.4% RUFL, 6.5 pounds of corn dry matter. And look at the next one. This is interesting. Ryegrass, okay, that I did in a study at Clemson University, where we harvested ryegrass at a very early, immature, early vegetative stage. And it had 4.6% dry matter, was double that of alfalfa hay or corn. And it only took 4.8 pounds of ryegrass dry matter to give the same amount of RUFL as 3 pounds of distiller's grain dry matter. Then you get into oil seeds, and they have a much higher RUFL content than the other basal ingredients. So 1.8 pounds of whole cottonseed dry matter, 1.5 pounds of whole soybean dry matter, and less than a pound of flaxseed dry matter to give the same amount of RUFL as 3 pounds of distiller's grain. So the point is, everything we put in front of the cow is supplying RUFL. And again, I believe how the RUFL is interacting with the microorganisms of the rumen is the key to determining which road it goes down, which pathway, trans 10 or trans 11. So we know the nutritional risk factors that we talked about previously are controlling de novo milk fat synthesis through its influence over the course of biohydrogenation. And in some manner, these risk factors are sending linoleic acid biohydrogenation down the trans 10 pathway. And not only that, but they're doing it in a way where they interact with each other in a manner that can sometimes make it very, very difficult in order to assess how that influence can be overcome. So next we want to consider how these risk factors, or the combination of these risk factors, may be signaling the microorganisms to shift in the wrong direction. That is, shift down the trans 10 pathway. I think the answer to that is how the RUFL interacts with the outer membrane of the bacterial cell. Bacterial cells are held together within a single membrane that surrounds a whole cell. They call it plasma membrane. Okay. That plasma membrane is made up of lipid, in particular phospholipid. Phospholipids have a head and a tail. One example of a phospholipid is phosphatidylcholine, where the head is choline, the tail is always fatty acids. There's 2 fatty acids in the tail, always, no matter what the head is. Okay. Now, notice how these phospholipids orient themselves in that membrane. The head's like water, so this is, they line themselves to the outside of the cell and to the inside, which is next to the cytoplasm or water inside the cell. The tails, which are the fatty acids, don't like the water, so they bury themselves between the head groups and try to stay away from water. So there's this very orderly arrangement of the heads and the tails and these phospholipids. Now, the membrane, this outer membrane of phospholipid, is very complex and is the vital lifeline of the bacterial cell, of our cells too, but ruminant bacteria as well, because embedded within this outer phospholipid bilayer are a number of proteins, like globular proteins here, helix proteins, integral proteins that are all different types of enzymes that perform vital functions for the microbial cell. Channel proteins that allow and facilitate the passage of required nutrients back and forth from the outside to the inside of the cell. And then there are various glycolipids and glycoprotein groups hanging on the outside of this membrane that act as receptors in order to attach essential nutrients and other factors that are, again, important to the lifeline of the cell. So if you do something that interferes with this outer membrane, it could have an impact on all these enzymes, all the movement of the acquired nutrients back and forth, this whole membrane function. So when ruffle comes into the rumen and is exposed to the microbial population, and remember that it's always coming in. It doesn't matter whether you're feeding any high-fat ingredients or not. Ruffle is present in everything you feed the cow. But if the ruffle levels are below antimicrobial, they're not causing any problems with the plasma membrane and its structure and its morphology. It's allowing all the enzymes in the membrane to function normally, and the cell is metabolizing the way you would hope it would at a high level. But if ruffle levels go above antimicrobial, and it may be the same level of ruffle, you don't have to increase ruffle levels for it to shift from below antimicrobial to above antimicrobial. Something else could happen that would cause the same level of ruffle to suddenly become antimicrobial. It penetrates within this phospholipid bilayer. It causes disruption and disorganization of the fatty acid tails that are in there, and this has negative impacts on the enzymes and passage of nutrients back and forth and so forth, and the cell does not function properly. So high ruffle has antimicrobial effects by disrupting the phospholipid bilayer of the microbial cell. Now, this is not a new concept. This is a review article that was published in 2018 that describes 4 mechanisms for how fatty acids express their antibacterial activity. Disrupting electron transport and uncoupling oxidative phosphorylation are both enzymatic pathways involved in energy generation, ATP synthesis. They operate more in aerobic cells. Now, since we're dealing with anaerobic environment, anaerobic microorganisms, more important to the rumen is the fact that when RUFL becomes too high, it can increase membrane permeability and leakage because it's disorganizing this phospholipid bilayer and disrupting membrane enzyme activity, and that can shut down or interfere with the function of the microbial cell. Ultimately, send biohydrogenation in the wrong direction. The interaction among nutritional risk factors that reduces milk fat is often explained by a shift in the microbial population community. I agree with that. It's been documented on more than one occasion that if you feed too much fat or too much starch or the combinations of other nutritional risk factors, that eventually you would see a change in the microbial population community. But that's further on down the road. And the trigger for that is the antimicrobial effect, which precedes the population shift. So what I want to show you is that these antimicrobial effects occur very quickly, far quicker than what you would expect to see an extensive change in the microbial population community, and also show that the antimicrobial effects selectively target the trans 11 microorganisms, allowing the trans 10 to take over. And finally, that antimicrobial effects are a summation of interactions among many nutritional risks, and thus explain the interactions that we see in the field. Now, how quickly, it's important, how quickly does RUFL in the diet cause microorganisms to express antimicrobial effects or to send biohydrogenation in the wrong direction? Okay. Here's a study from Microbiology Journal 2017. It shows the metabolic pathway intermediates in Butyrovibrio fibrosolvens. This is a major microorganism involved in biohydrogenation. And this study measured the amount of metabolic pathway intermediates. So the more intermediates, the more that the cell was functioning the way it should have. So this was the level when there was no linoleic acid. This was the level when they put in 0.2 milligrams of linoleic acid per ml into the culture media of the microorganisms. 0.2 milligrams per ml. That is a level very, very easily achieved with just the basal RUFL supplied in your TMR if you don't have any other fat. With 20 minutes, in only 20 minutes, the metabolic pathway intermediates were reduced 95%. What does this mean? It means that in a very, very short time, very, very quickly, the microorganisms were bothered by the presence of the linoleic acid to the point where it shut down metabolism. There's a 95% drop in cell metabolism after just 20 minutes of RUFL exposure. So you go too high on RUFL, the microorganisms in the rumen are going to know it very quickly and going to respond very quickly. Now, I look in the laboratory in continuous cultures at how quickly I got a trans 11 to trans 10 shift. Okay. So I ran continuous cultures for 12 days. The first 4 days, I had no added fat. And then on day 5, I introduced a dose of free oil, soybean oil, to intentionally increase RUFL and send the pathways in the wrong direction. How quickly did they change? I took samples every hour after I introduced the high RUFL. And you can see that introducing the RUFL cause both the trans 10, its scale is on the right, and the trans 11 to increase. You see the trans 11 is much higher than the trans 10. That's normally the way it is in the rumen. They both increased, but the pattern of increase is different. The trans 11 is increasing, but it's increasing at a diminishing rate, whereas the trans 10 is increasing, but increasing at an increasing rate. Trans 10, within just a few hours of having this dose of high ruffle, continuous cultures of rumen contents, the trans 10 is beginning to take over. Now, not all microorganisms are affected by linoleic acid the same. Now, some microorganisms in the rumen carry out the trans 11 pathway. Some carry out the trans 10. Here's a listing and the example citations that have demonstrated the microorganisms that are responsible for using linoleic acid to go down the trans 11 pathway and the ones that go down the trans 10. Linoleic acid. So do fatty acids exhibit antimicrobial properties for trans 10 differently than for trans 11? Here's a study that examined that question. They took all these rumen microbial species, all known rumen microbial species, and they measured the amount of enzyme disruption from linoleic acid using specialized fluorescence techniques. They were able to measure how well that phospholipid bilayer was disrupted and disorganized by linoleic acid. The farther these bars go to the right, these black bars, the more disruption occurred from linoleic acid. So you can, as you look down the line here, you can see that not all these microorganisms were disrupted by the same degree by linoleic acid. So when you put a certain amount of ruffle coming in from the diet every day and it gets into the rumen and interacts with the microbial cell, that ruffle could be disrupting the membrane activity of some microorganisms more than others. The one that's disrupted the most up here was on the list on the previous slide as going down the trans 11 pathway, the good pathway. The one on the bottom down here was on the previous slide I showed you as going down the trans 10 pathway. So it's one example where linoleic acid interfered more, had greater antimicrobial effects with a trans 11 producing microorganism than one with a trans 10 microorganism. Here are a couple of examples of how antimicrobial effects of a certain level of ruffle can be modified by interacting factors. In recent studies published in abstract form in 2018, they showed that low pH enhanced the antimicrobial effects that linoleic acid had on a fibrosolvent species. Fibrosolvents follows the trans 11 pathway, not connected to lowering milk fat. So low pH enhanced the antimicrobial effects on that trans 11 pathway, but it did not have that same effect on the trans 10 pathway for an alternate microorganism. So it explains how pH is an interacting factor, enhancing the antimicrobial effects of a certain amount of unsaturated fatty acid or ruffle on microorganisms involved in the trans 11 pathway, but not trans 10. Another example is lactate. Lactate has been shown to have a direct effect of accentuating the antimicrobial effects of unsaturated fatty acids on the trans 11 fibrosolvents. That was work that was done published in 2010 in a microbiology journal. So that would explain starch in both of these cases. Starch if it lowers pH, and starch if it leads to the production of lactate. But pH alone, through the use or absence of buffers present in the diet, could have an effect on this as well. Based on the microbiology results that we talked about, I think I can suggest a sequence of events that occurs in the rumen that explains the interaction occurs among these nutritional risk factors. Let's start with a situation where you have a certain amount of RUFL coming in from the diet that's not causing any problems with the microbial population. It has very low antimicrobial effects. This could be RUFL simply from basal ingredients, or it could be RUFL from basal ingredients plus some added fat sources. But whatever the source, they're not causing problems with the function of the microbial cells. And then there's a change that occurs in the rumen environment. Maybe pH is lowered for some reason. You change starch levels or the rates of degradability of the starch. Yeast starts coming in, or there's a change in your program for ionophores or overcrowding and so forth. But there's a change in one or more of these other risk factors that could be very, very subtle. But those subtle changes in those other nutritional risk factors can now all of a sudden cause that same level of RUFL that was causing no problems before to suddenly become antimicrobial. So now that level of RUFL can now interfere with the plasma membrane of select species of microorganisms in the rumen and interfere with the organization and structure of the plasma membrane. As a result, the enzymes and function of that membrane are impacted negatively, and they don't function so well. And from what we've seen from the microbiology results, this has a greater effect on shutting down the trans-10 or trans-11 microorganisms than it does trans-10. The trans-11 microorganisms that carries out what we call the good pathway of biohydrogenation is impacted much more by the antimicrobial effects of the RUFL. So they're now not using so much linoleic acid. Linoleic acid is now diverted away from the trans-11 microorganisms and made available to the microorganisms that utilize the trans-10 pathway. And as a result, more linoleic acid going down trans-10, we have more of the trans-10 cis-12 CLA that causes milk fat to go down. So the bottom message here is a certain level of RUFL that can be totally harmless in some situation can now become a problem when you have one or more of these interacting factors that become available. This is a statement that was taken from a 2018 publication, Frontiers in Microbiology, that kind of sums this up. It says, all the results from these microbiology studies suggest that when the trans-11 pathway is lacking or is compromised in some way, the trans-10 pathway must then do the biohydrogenation of unsaturated fatty acids. It says the rumen is destined to carry out biohydrogenation. And when its primary pathway, and its primary pathway is the trans-11 pathway, not connected to milk fat problems. When that trans-11 pathway is compromised, it has a backup. And the backup system is trans-10. And all the nutritional risk factors we talk about compromise the trans-11 pathway more. And as a result, it shifts biohydrogenation to trans-10. Now, given that information, we still have to manage these nutritional risks. And first and foremost risk factor is to be careful not to overload the rumen with unsaturated fatty acids. If there were no other risk factors present, we still wouldn't want the ruffle to become so high as it interferes with the function of the trans 11 pathway and sends things down the wrong road. Now, so how do we do that? Well, we've already taken advantage of the fact that we know that saturated fatty acids are not the problem here. They do not express antimicrobial effects. So for feeding bypass fats that are high in palmitic, stearic, or a combination of the two of them, there are not going to be a problem with the course or the direction of these biohydrogenation pathways. So the focus is on ruffle, like we've been talking about. And we don't want to get away from feeding ruffle. There are many, many fat sources that are their prices go up and down, and that makes them very economically attractive at times. And we want to be able to go to them. We just have to limit the amount we use. Distiller's grain, cottonseed, tallow, any other products that may come along with high fat in them, waste products, bakery waste, and so forth. At great prices. We want to be able to use them, but we just have to stay below the antimicrobial line. Now, I'm diverting more and my attention to the fact that all ruffle is not created equal. There's low risk and high risk ruffle. And low risk ruffle would include fatty acids coming in from hay, unprocessed corn and corn silage, whole oil seeds, and calcium salts of fatty acids. In these cases, the fatty acids are still going to be subjected to biohydrogenation, but it's going to be metered out more slowly so that it doesn't pile up onto the microbial membrane all in one pulse dose, cause problems. So higher risk ruffle then are processed or over-processed corn and corn silage, where everything is opened up, allowing the internal fatty acids to come in contact with the microbial plasma membrane all at once and pile up on it. Grinding oil seeds, free fats and oils, and distiller's grain are all examples of high-risk ruffle. So I don't try to tell customers how much ruffle is too much, because the bottom message of our whole discussion today is that the amount of ruffle that becomes a problem varies according to the presence of all the other interacting risk factors. So there is not a single number in how much ruffle is safe. But what I can do is tell customers when their ruffle is high so that they can pay attention to that. Now, to get an answer to that, I got the help of the folks at Cumberland Valley, and they gave me data from all of 2018 showing the relationship between the frequency of ruffle they found in dairy TMRs. We selected out the dairy TMRs by filtering the data to include only TMR analysis that were in the 15 to 20% protein range and 28 to 35% NDF. Did the best we could to select out beef and other diets. So that gave us 2,592 observations. The average ruffle was 2.46. And I have it broken down here in a half unit ruffle increments. As you might recall previously, I talked about it in laboratory studies, continuous cultures. We saw a big increase in ruffle. Not ruffle, but we saw an increase in transcensus 12 when the ruffle got to 3 or higher. So I kind of use that as my alarm point. When the ruffle is above 3, my alarm goes off that I'm on the high side and there's a potential risk of causing antimicrobial effects in the rumen. And in this data set, 16% of all the TMR's analogs have ruffle greater than 3%. So if you go onto a farm, they're experiencing fat levels that are below what they normally have or what they expect they should have, take a look at their ruffle. That probably should be the first thing that you do. And make sure that they're not on the real high side of feeding unsaturated fat. If they're greater than 3%, they're in the top 84th percentile. Okay. There's only 16% of the herds across the United States that are feeding that level of ruffle. 84% are below that. At least gives you a guideline of whether they're on the high side or the low side. But again, it's not an indication that that's too much ruffle. We need to continue to pay attention to pH as a risk factor. Okay. And there are a multitude of factors that can help here. Remember our discussion with the microbiology, pH and starch and lactate and all that very much determined whether or not a certain level of ruffle was going to be a problem, become antimicrobial. So we want to focus, can we maintain effective fiber and adequate rumination so we have good chewing and good salivation and buffering. Don't let the starch rate of degradability get too high. I've seen this happen with high moisture corn, especially with corn silage. Some cases, rates of degradability of starch were greater than 85%, almost explosive in the rumen. That could lead to lactate and low pH, which can affect antimicrobial effects. How are you using buffers? Many go to a combination of bicarb, potassium carbonate, or sodium carbonate as a way to even out and reduce the variation in pH that goes in the rumen. Pay attention to feeding management. Are you mixing the TMR well? Don't overcrowd. Make sure push-up is occurring often enough, and feeding frequency can have an impact too. Feeding frequency, why? Because feed more, we're metering out ruffle over a longer period of time. Feed once a day, that big dose of ruffle is coming into the rumen all at once and ramping up the levels of ruffles that can interfere with the microbial function. Here's a relationship that I looked at between starch levels in TMR and change in milk fat percent. You can see the negative relationship. As I increased starch, there was a steady drop in milk fat percent. Now, this was among studies that fed high-risk ruffle-free oils. And in those studies, there was for every 0.1 increase in starch, there was a 0.05 drop in milk fat. So another way, 0.2 increase in starch levels when there was high-risk ruffle present resulted in a 0.1 drop in milk fat. Now, if your ruffle is low risk, this is not going to apply. But it demonstrates the starch interaction that occurs when you're trying to feed a certain level of ruffle. Other risks can come along. And although we don't have verified, clarified scientific data on the effects of wild yeast on the pathways of biohydrogenation, we often see it in the field. Oftentimes, when yeast values are very, very high in corn silage, they're experiencing milk fat problems. My alarm goes off when I see yeast counts in silage approaching 1 million CFUs per gram. And I was on farms recently that had 50,000 CFUs or 50 million CFUs. And I've been on farms where I've seen 100 million CFUs. And they all have been dealing with milk fat problems. Ionophores. They're an interacting factor. I think alone not a problem, but it intensifies the risk when other proper feeding guidelines are not followed. So if we're already interfering with the function of the microbial membranes by feeding too much ruffle, the presence of ionophores, which also has an effect on microorganisms, is not a positive factor. It continues that interaction in a negative direction. Now, finally, my final statement is we look to get help from supplements that boost up milk fat, and they have been providing tremendous benefit to us, such as high palmitic bypass fats. More recently, there's been some focus on MHA and how it may be used to help boost up milk fat. But I do want to caution us that these are not a substitute for managing the trans 10 risk when the milk fat is low. They're kind of like Band-Aids to cover up the fact that we have a rumen imbalance going on and we have a combination of factors that's interfering with the microbial population and sending things in the wrong direction. We want to continue to use them, but we want to, at the same time, we want to also not take attention away from the fact that we have other risk factors at work there that we need to manage. With that, thank you very much. I again appreciate very much the opportunity to have participated in this great webinar series.
Thank you very much, Tom. Maintaining strong components is critical in this dairy environment. I hope you listeners found valuable recommendations in the information he presented. Shortly, we will switch over to the live part of the webinar to take your questions. Before I do, I'll introduce our next webinar and thank our co-hosts and the webinar sponsors. Next month, on October 10th, we will be joined by Dr. Michel Watteau from the University of Wisconsin-Madison. His talk will focus on the carbon footprint and methane emissions in dairy cattle. Dr. Watteau grew up on a dairy farm in Belgium and has a strong commitment to making farms economically profitable as well as more environmentally friendly. Given the concerns we all have about shifting climate patterns and the role that agriculture may play in human factors influencing that shift, we are very pleased to host both Dr. Watteau this fall and Dr. Frank Mitlower at UC Davis, whose Twitter name is GHG Guru, next year for a discussion on the effects of dairying on climate change. We hope these presentations from these noted researchers will open a healthy discussion and awareness of the facts related to the industry. As part of the talk next month, we will be featuring information on climate impacts and amelioration strategies on our blog and social media pages during the month of October. As with this webinar, there will be 2 opportunities to join: 9 a.m. Eastern Daylight Time and 6 p.m. Eastern Daylight Time. I am taking this opportunity to thank my co-hosts and the people who make this possible. The webinars are organized and produced by AMTS USA and Global. Our longtime collaborator is Paula Torillo of Afino, which hosts the series as El Webinar del Nutritionista. She receives support from Tecnal, Vinis Claden, and Guillermo Lerman and RR Lab in Argentina. Marcelo Hens Ramos, AMTS distributor and the director of 3R Lab in Brazil, delivers the webinar for our Portuguese-speaking audience. Our AMTS distributor and the director of AnsiTech, Sean Lee, hosts the Mandarin language webinar platform. We are joined by our AMTS distributors Vadim Bekchepnikov of Novo Lab in Russia and Elena Bonfante of Dairy Innovations Italia of Italy. We are particularly grateful to the generous sponsors who make it possible for us to get great speakers and manage the program. We thank our gold sponsor, Arm & Hammer Animal Health, makers of cattle feed ingredients that optimize dairy cow health. Our silver sponsors are Ajinomoto Heartland, superior nutrition through amino acids, makers of AgiPro L, Dairyland Laboratories, Virtus, makers of Strata with EPA, DHA, omega-3s, and Prequel with omega-6s, Dairy One Forage Laboratory, and Jeffo, Life Made Easier. Our bronze sponsors are AminoMax, Purdue Agribusiness, Adaseo, Origination Inc., Novo Meal, Chemin, and PMI. Each of these companies support education and research worldwide. We hope that you consider them in your formulation decisions. I will now open the floors for questions. English language listeners, I will read your question. Remember to type your questions in the Q&A tab or the chat window. Hopefully you can now see the slide set with our sponsors, and I'm going to temporarily unmute both Elena and Dr. Jenkins so that we can all say hello. So hi Elena, hi Tom.
Good morning.
Hi,
Excellent. Now, if anybody out in the audience, if this is not coming through, because I can hear everybody, go ahead and shoot me a quick chat in the chat window, just so that I know. In the meantime, I'm going to get Tom started on the first question. If I add propionic acid to stop corn silage heating in the bunk, will the wild yeast that were initially causing the heating still affect the BH process or not?
Very good question from out there. Unfortunately, we don't understand the science behind how yeast become such a risk factor for milk fat. I see it all the time, but we don't have the science behind it yet. Anything that we can do to limit yeast is going to be good for the cow. I haven't found any good things that come out of a lot of wild yeast growing in fermented feeds yet. So certainly one of the good things for limiting is that we help support higher milk fat. So if you, I would prefer that you limit yeast growth on the bunk side of things, or in the bunks. But if it's in front of the animal for long periods of time under severe weather conditions, you could have spot growth that way. And that might have an effect on the course of biohydrogenation and milk fat as well. So I certainly think it certainly couldn't do any harm, and probably good to make every attempt to try to limit the yeast growth that's occurring in these feed we put in front of animals.
Okay, thank you for answering that question, and thank you for asking the question, person out there. Elena, I'm going to see if you have questions. I do have another question, and that's what I have in my chat window, but do you have some questions to ask?
yes, Marianne. I connected to the question about the propionic acid, and actually I was wondering, because now here in Italy, what I think all over, it's getting very popular, the Buchneri, in inoculant, in order to have an acetic fermentation, okay? And so I was wondering, having more acetic acid in the forages instead of lactic acid, can that have an effect also inside the rumen somehow, or doesn't matter?
Elena, I think the question was if you have more propionic acid instead of lactic acid, is that beneficial? Is that correct?
Yeah, acetic, sorry. With
Acetic.
Buchneri, you increase acetic fermentation.
Yes.
Acetic.
Yes. Okay. The acetic acid would be good. Lactic acid would be bad when it comes to biohydrogenation. Of course, we push the limits of starch feeding when the price of corn in the U.S. is favorable to do that, and that pushes us on the edge sometimes of lactic acid. So we want to feed starch, and we sometimes push the lactic acid thing in order to get there. And when we do, what I was trying to demonstrate today, lactic acid kind of opens the door up on these microorganisms, opens up the membrane door, and lets this ruffle come in. And once the ruffle gets into the membrane of these microbial cells, then that's when the whole process of damage starts. So if we can, lactic acid in that sense is not good when it comes to biohydrogenation. Acetic acid would be good. Acetic acid is the main fuel for the mammary gland to carry out de novo synthesis. And there's no reason to think that acetic acid has any kind of harmful effects on the process of biohydrogenation. So limiting lactic acid certainly would be a benefit.
Okay, so also when we are talking about the acid that is it formed during the fermentation of the silages?
Oh, acetic during the formation, during silage fermentation.
Fermentation, exactly.
Yeah, okay. Yeah, well, good, we want good silage to be stabilized, and it takes low pH to get down there in order for it to get stabilized. And I don't, and we need lactic acid to do that. But I don't think we're talking about enough lactic acid coming in good silages in order to open the door up that much on these microbial plasma membranes as to cause problems. So I wouldn't want to say that we want to try to avoid lactic acid in the silage making process, because we need that to drop down the pH. But it's only in the rumen that we would then want it to not get out of control from starch fermentation that could lead to problems. So we need good silage. Follow our good silage making rules that we've been taught over all the years, and manage, yeast to a minimum, and packing well and keeping oxygen out and that sort of thing are all going to lead to good milk, supporting good milk fat also.
Okay, thank you very much.
Okay, thank you, Elena, for asking those questions. I have a question from Rolando, and it is, what do we do with inert fats that have linoleic in high amounts? Can we use them?
Okay. Thanks, Rolando, for your question. Yeah, well, the key part of the question is, Rolando says they're inert fats that have high linoleic acid. So he used the term inert fats when he described them. So the assumption here is that they are fats that do not interfere with the rumen fermentation process or the function of these microorganisms. And there are some commercial bypass fats out there that have higher levels of unsaturated fats. And the reason they have them is because they are trying to supply more omega fatty acids to the tissues of the animal, thinking that the animal may not have enough of those coming in. So there's good reason why these fat sources commercial fat sources are higher in unsaturated fatty acids. Now, protecting unsaturated fatty acids from causing problems in the rumen is a tough job. It's not easy to do. There are some calcium salt products that do it, and they work very well in most situations. They can cross the line. We can create conditions in the rumen where those commercial bypass fats would release the unsaturated fatty acids. As examples would be if the pH in the rumen were to become very, very low. In the case of the calcium salt, then the calcium is released from the unsaturated fatty acid, and that puts the linoleic acid then in a position to cause these antimicrobial effects. So if you're managing the diet very, very well and following good nutritional management practices, then these bypass fats with unsaturated fatty acids are probably going to work very, very well. But again, there could be situations, you could put the cow into situations with your feeding management that would cause these bypass fats to not work very well. But usually those are situations that we try to avoid. So a long explanation, but the answer to the question is inert fats that have high linoleic acid can work, have worked well. And again, when you say high amounts, we don't want to push that too far. But usually when you're trying to supply unsaturated fatty acids, they're usually only needed in lower amounts. You don't have to feed a lot of these to get the amount of unsaturated fatty acids you need. So I hope that helps, Rolanda.
Thank you. And if we have, yes, Rolanda says thank you so much for the answer. Unless Elena has some more questions, we probably don't have any more questions for this morning. I haven't seen any yet in the chat window, and I'm out of questions in the Q&A. I did have some, in doing a little bit of research, preparing for the webinar, most of the recommendations always seem to hinge on do your best with your forages and almost more preparatory methods. Wondering what are your best recommendations when somebody's not got the best quality forages, or they're dealing with forages that maybe are driving down the components. What can they do at that point if they can't get out of that sort of situation?
Yeah, that's a tough one, but it's very relevant. It's a great question, and it certainly implies in many parts of the world, and including parts of the United States as well. Forage quality can have, low forage quality definitely has an impact on milk fat. And while we always encourage people to try to boost up their forage quality program, that's not always very easy or reasonable to do. Part of the problem with the low forage quality, like I alluded to earlier, was with the lack of fermentable carbohydrate, you may have a lack of supply of the acetate building blocks coming from the rumen in order to support a good level of de novo milk fat synthesis. So you need something fermentable in there. And you are going to have to look for whatever grains, byproducts that are available and economically available in your particular situation that would help to support VFA synthesis and production in the rumen. Now, the forage offers more than just that. It also helps to provide attachment sites for fat so that it doesn't come in contact with the microorganisms as well. Good forage particle size is an additional asset on that. So it's going to depend on providing some sort of larger particle size fibrous and non-fibrous carbohydrate sources that could take the place of missing out on these forage quality advantages that are often. But that's a tough question for me to answer. I'm sure some of the other viewers would be able to chime in and help on this, but just need to look for what other fermentable carbohydrate sources are available to take the place of them.
Thank you. Thank you. Let's see, I have a couple questions. Is the contribution to the risk of milk fat depression in the individual fatty acids in RUFL equal between the three?
Yes, I often get that question. And you can see how I calculated that I'm giving equal value to all three of them. Actually, the data that's been generated on the individual fatty acid effects on antimicrobial effects show that they're not equal. That the more double bonds that are in the fatty acid chain, the greater the antimicrobial effects. So oleic acid has more antimicrobial effects than stearic acid has, linoleic more than oleic, and linolenic more than linoleic. And that kind of fits in with what we've seen in the fat feeding field data studies as well. We can still see that oleic acid causes milk fat to go down, but at an equal level, at equal amounts of linoleic acid cause it to go down more. So I don't think they're equal, and I'm continuing to play around with the Ruffel equation to try to find how its correlation with the generation of trans-10 cis-12 CoA in the rumen can be made better by modifying the calculation of Ruffel.
Okay, thank you. Another question. Where does the high Ruffel content in forages come from?
Well, plants in there, of course, it varies among forage species. But plants in their very early vegetative stages synthesize a great deal of fatty acids. And it's mostly in the leaves, and it's mostly in the chloroplasts of the leaves. So as the plant matures and you have leaf loss and becomes more stemmy, the fatty acid content will go down. But it's sometimes surprising to see how high the unsaturated fatty acids can go in some of these grasses, especially temperate grasses, at very early emergence, early vegetative stages. And it comes from, it's just the normal energy supply within the plant that is put there in early vegetative growth and then disappears. It doesn't disappear, but goes down as the plant matures.
Okay. Thank you so much, Dr. Jenkins. I have a couple questions that came from Sean Lee. And his first question is, one of the areas that you discussed in the presentation is the trans 10 versus trans 11 pathways. These, I think that's what T10 means. These are two pathways that are covered or saturated linoleic acid into, convert saturated linoleic acid into stearic acid. How important is a T10 pathway to the saturation of oleic acid and linoleic acid by rumen bacteria?
Yeah, the oleic acid does, despite earlier descriptions of biohydrogenation several decades ago that showed it going directly to stearic acid, that has been debunked because I've done studies myself to trace the carbon flow of oleic acid with stable isotopes and was able to show clearly that carbons from oleic acid do flow through trans intermediates. Now, they don't yield CLAs. Oleic acid does not directly yield any CLAs, but it does yield trans 18-1 isomers, including some trans 10. Now, the prevailing scientific evidence seems to suggest that the trans 10 is not a direct cause of low milk fat, that it's a CLA. The trans 10 is just kind of a marker for that side of the pathway. So the way we think oleic acid operates to lower milk fat is by indirectly causing antimicrobial effects on the trans 11 microorganisms, the way I've described today. And by causing these antimicrobial effects, then it's shifting the pathway of linoleic acid biohydrogenation. Now, linolenic acid, or 18-3, is still a controversial subject on whether or not it directly yields CLAs, in particular trans-10 cis-12. My isotope studies have suggested that those microorganisms have that metabolic capability to do it. Biologic capability. It doesn't mean that it's done all the time under all conditions, but my tracer studies did show they had the biologic capability. And this was just recently confirmed in a study coming out of Spain where they labeled linolenic acid, again with stable isotopes, found the isotope distributed among 40-some isomers that included trans-10 cis-12 18-2. So linolenic acid may directly produce CLAs on the trans-10 side, but also definitely causes antimicrobial effects that could also cause linoleic acid to go down the trans-10 pathway.
Okay, so when Sean sent this, he CC'd Tom Taluki and Bill Mahana. And he said, so some of the responses over the weekend from Bill was, he said that from Fermentics analysis, we know there is a balance between starch KDs and fiber KDs. Further, too rapid of a starch. Some of these words I'm not familiar with, and I don't know whether I'm going to put it in the chat for you, Tom, and you can assess this and comment on it. In the meantime, I am going to ask a question from Carlos. We have some better modification or better explanation of that question that he was asking earlier. He says, our feeding system determines a certain level of fluctuation in milk fat concentration. So he would like to know, what is the minimum fat level at which you should suspect there is a high RUFL problem? Is that better?
Yeah, I think so. Yeah.
Okay.
I think Carlos is asking, what is the minimum amount of fat you could add to the diet to have an acceptable RUFL that's not going to cause milk fat?
Yes, and if that is what he's asking, Paula will probably confirm with him. And this is in a partial mixed ration, it's not a TMR, it's a PMR.
Yes, yeah. And that goes back to the one that we were discussing earlier when they asked what is the right amount of RUFL to feed? What is the best amount to feed? And again, I answered that, was saying that that answer fluctuates depending on the other risk factors, the interacting factors that occur. So I have used some guidelines in the past for feeding unsaturated fat sources that have taken into account the level of unsaturation in the fat and the amount of fiber in the diet to give me some guidelines to limit the amount of unsaturated fat I feed to try to stay out of trouble as much as I could. So again, you can't give one answer for that. Many times you do hear, you hear, oh, don't go over 7% fat, or 1/3 from basal added and bypass and so forth. But those are oversimplistic and don't reflect the fact that the damage that those fat levels can cause vary depending on the complexity of the other risk factors. So keeping it low, if you're not sure what to do, you keep it low, and you add a half a pound of an unsaturated fat source to start. If things go well, then you boost it up from there on the conservative side. But the success of that and the level that's achieved, the successful level, is going to vary from one place to another because the ration is different and has different risk factors in it.
Okay, thank you for handling that. I have a series of questions from Marcos to give Paula some time to work on translating on her end. And then we'll try to circle back to, I put in the chat window to you what Bill Mahana said, but we'll first ask some questions from Marcos. His first one is, considering that whole cotton seeds are a slow release fat source in the rumen and that it usually increases milk fat content, do you think the RUFL in this feed should be counted as RUFL for ration balancing?
The RUFL is slow released, is consistent with the higher milk fat, I agree. And then what was the second part of that?
And that it usually, let's see, it's a slow release fat force and that it usually increases milk fat content. Do you think RUFL in this feed should be counted as RUFL for ration balancing?
Oh, I see. I get what they're asking. I think what the question is, can I eliminate or delete the RUFL from my RUFL calculation if I think that it's slow release, low risk RUFL?
Okay.
Yeah. And I'll tell you the reason I continue to use RUFL is one simple strategy. I want to know all the fat a person is feeding from all sources, and I don't want anything left out. I don't want hay left out or silage or any byproducts or so forth. So I still want to know a RUFL that includes everything in the diet when I first am troubleshooting why milk fat may be lower than what a nutritionist is looking for. Then, if I see that the RUFL is high, then I ask the question, how much of that do I consider to be low risk and how much of it be high risk? If the majority of the RUFL is high risk, then I know to go in and start trimming back on some of the high risk RUFL sources to try to help with the problem. If I think that it's largely low risk RUFL, then I turn my attention to some of the other interacting factors. So if I understand the question correctly, I don't want to modify my RUFL calculation to leave anything out. I still want everything in it from everywhere, so I know the total amount that's been contributed by everything, and then go on from there to try to assess the degree of risk.
Okay. I'm not fully certain that Marcos can hear to redirect us if that wasn't what he intended. And I see that he's left again. I think he's having issues with his sound. Second question from Marcos. Could you comment on feeding of high palmitic acid fat sources?
Yes. High palm fat sources have found very good consistent success in elevating milk fat. They do this because they are providing C16 fatty acids, that's what palmitic acid is, to that mixed fraction. And by boosting it up in the bloodstream, the mammary gland is able to extract it out of the blood as a preform for the preformed portion of the mixed. And that's been successful in raising milk fat 2 or maybe 3 points. There's a lit to that. You can't continue to increase palmitic acid to much beyond 1.5% of the diet and get milk fat to go much higher. And as I pointed out in my last slide, if you're getting into milk fat problems, palmitic acid is going to help you give some quick relief, but you need to continue to final statement is work on finding the source of the trans 10 problem. You can see on this slide here, it's brought up
high palmitic bypass fats. More recently, there's been some focus
so
on
for some
MHA
reason we're running
and how
the video
it may
again.
be
We need
used to help
stop
boost
something.
up milk
Oh,
fat.
I know what it is.
But I do want to caution us that these are not a substitute for managing the trans 10 risk when the milk fat is low.
Let me get to where it's not talking. There. Okay.
Well, that actually worked out well because I said again on that slide what needed to be repeated.
Yes, thank you. Thank you. I'm not sure people could hear it as well. Let's see. Next question is it necessary to have ruminal microbial adaptation to fat feeding? The biohydrogenating capacity of the rumen can be changed by the biohydrogenating capacity of the rumen be changed by previous feeding of fat? Is fat, if fat is to be fed after calving, would you also give fat pre-calving? Is there any evidence that supports it?
Okay, that's very good.
There's a lot wrapped up in that.
Yeah, and it's a very good question. And it's not one that I have a direct answer for from structured research trials. But what comes to mind right away when I heard the question was, I've done a lot of studies where I've suddenly introduced fat into the diet and can see a shift in the trans 10 to trans 11 pathways very quickly, as I showed today. Also studies where they were fed fat for a while and then taken off of it and then put back on, and they still responded the same way. So I haven't seen much evidence that pre-adapting them, exposing them to RUFL has given them any advantage in avoiding sending linoleic acid carbons down a particular pathway. Now, as far as the feeding fat pre-calving versus post-calving, I agree that introducing fat into the diet pre-calving, first of all, I think the main reason to do that is reproduction. That's the main advantage. Some fats can provide an advantage on the repro side. But aside from that, I think there is an advantage of letting the animals become used to some fat source, especially bypass fats, that they never might have seen before, and ramp them up slowly on those and get them kind of running more smoothly once they go into lactation.
Okay, thank you. Let's see, moving along, because I'm so grateful that Marcos is joining us again, because he does ask these fantastic questions. His next question is there any feed additive with potential to favorably manipulate ruminal lipid biohydrogenation, for example, capable of stimulating T11 metabolites or complete hydrogenation?
No, we're looking for those all the time. Right now, all we can do is just manage the bad things so it doesn't go down the bad side. We don't have anything at our disposal right now that ramps up the good side, the T11 side, which would work very, very nicely because it would help to ensure that linoleic acid carbons would be going down the trans 11 side of the pathway and continue to rob the T10 pathways of the linoleic acid they needed to function. That would be very nice, but not aware of anything that does that at the present time.
Okay, next question from Marcos, and I think he's probably pulling his hair out at this point. Do jerseys respond, let me see where to go. Do jerseys respond more in milk fatty acid profile and fat percentage than Holsteins? Are they better experimental models to evaluate the effects of dietary manipulation on milk solids? And I'll just throw in I'm always partial to jerseys, so yes, they're better at everything, but how about a scientific take on that?
Yeah. Your jersey enthusiasts seem to be everywhere. And the question is, are they more sensitive? I think making them a better model. Their rumen microflora certainly carry on the pathways of biohydrogenation the same way that Holsteins and other breeds do, and the same two pathways exist. And Jersey milk does differ. Jersey milk fat synthesis does differ from that of Holsteins in several ways, but they seem to respond very, very similar to the risk factors. Now, I think there's a little bit of difference in jerseys that may modify those risk factors, such as eating patterns, feed selection, things of that sort, that might make them a little bit less susceptible to a certain risk factor causing a shift in the trans 10 to trans 11, but the pathways are still there. The same risk factors apply, but there's a little bit of difference in their eating patterns and the flow through the rumen that I think might give them a bit of advantage at times. In fact, I have a study going on right now at Clemson in cooperation with other Clemson faculty where we're comparing Holsteins and Jerseys on that exact question.
Oh, neat. I'll watch for that. Let's see, another question. Could you comment on fat feeding around calving plus or minus 21 days relative to calving?
Yeah, I think that's similar to the other question of Marcos about
yes,
pre versus post calving feeding of fat. The advantage, like, just to quickly go over that again, the advantages of minus 21 days, in my mind, are a reproduction advantage if you're using unsaturated fatty acids that have been shown in a number of studies to have positive effects on reproductive performance of the animal and immune function as well. That's not the topic for today, but they have been shown to be connected to those advantages. So the other one is, there's some talk, a little bit of talk out there about guiding the fetus through fat feeding in order to perform and function in a certain way in early life through first lactation.
Yeah, I think that would have been some of, well, let's see, we had some fetal programming discussion in regards to beef cattle. And then a couple months ago, Jeff Dahl talked about heat stress. So I can't see why other things like that wouldn't do some fetal programming and switching.
Yes, very good. And the fetal programming has been shown to exist in a couple studies where the feeding different types of fat pre-calving has caused the animals to perform differently post-calving, even when they weren't fed any fat at all post-calving. So it does exist in dairy as well.
Yeah, I was, we did a beef webinar series last year, and I felt like the beef studies have not been as particular as dairy studies have been yet, because there isn't the economic rewards for it in terms of, until you get to feedlotting them.
Right.
So
there's a beef nutritionist at Clemson who I visit with regularly, Dr. Nathan Long, who has done some work on fetal programming on the beef side, and he has exciting prospects for what can be accomplished and done, but I don't know about the economic value of it.
Right. It was Alejandro Relling who spoke on it, and he said it's there and they've proven it, but in order to make it economically viable, those animals going through the feedlot have to be somehow identified as potential for better and quicker growth, and that just isn't there right yet.
Yeah. So that's another example of reality keeping pace with science advances.
Yes.
Yeah.
I have a question. Oh, sorry, go ahead.
Yeah, I was just going to end it by saying that sometimes the best of the scientific advances in information kind of get lost because it's difficult to carry them out under our current feeding systems. Yeah.
Yes. I have a question from Nestor. Can the bypass fat particle size, for example, in calcium soaps, be a risk factor for biohydrogenation and therefore lower the percent of milk fat? Is there a recommendable size for these products?
Yeah, there are commercial calcium soap products that try to take advantage of larger particle size in order to provide some shielding, some additional shielding of the oleic acid that's in there. Oleic acid is the primary unsaturated fatty acid in calcium salts of PFAD. And that could be an issue perhaps in situations, or an advantage in situations where the calcium salt might be at risk because of very low rumen pHs or something. Now, when the conditions are good in the rumen, there's probably not that much advantage to particle size because the release of the fatty acid, the oleic acid from the calcium, is probably not extensive enough to cause a problem with milk fat. And over many decades of feeding calcium salts of palm fatty acid distillate, we have very, very few instances where milk fat went down. So that's not a consistent problem. But if you're feeding in situations where your rumen pHs might be very, very low, that additional shielding could provide some advantage.
Okay, thank you. I'm waiting to hear back from Paula Alanis to see if she has any more questions. I do not have any questions. She'll let me know shortly. And I'm trying to see if there's anything else that I had from any of my other questions. I don't think so. Just to let you know, you had kudos from this morning's webinar. Harry Bristol said you did a great job, and you explained things well enough and slowly enough that he could understand. So hopefully everyone is enjoying that. There are no more questions from Paula. So I know you've had a long day. You're not joining us from South Carolina, but rather from California.
Yes.
So there's still a few daylight hours for you left to enjoy.
That's right. Thank you very much for having me. It's been my pleasure, and I'm a big fan of this webinar series, and I'm thrilled to have the chance to be part of it.
Well, we're very pleased to have gotten you to speak again. So have a good evening, and thank you everybody who joined us. All right. Goodbye, everybody.